This Week in Virology, the podcast about viruses, the kind that make you sick.
From Microbe TV, this is TWiV, This Week in Virology, episode 726, recorded on March 2nd, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 36 Fahrenheit, but it was quite cold this morning. And what accompanies that is a perfectly cloudless, bright blue sky, really sunny and nice.
I'm happy to be out of February because I find it really hard to pronounce. I'm glad we're in March. Also joining us from Austin, Texas, Rich Condit.
Hi there. 64 degrees. Brilliant sunshine. It's nice. I'm procrastinating, going outside to pull weeds. Tomorrow will be a better day for it. And I was going to give you trouble about your pronunciation of February, Vincent.
But, is that how I say it?
I restrain myself. No, you say February.
Yeah, because the February.
You drop the R.
It sounds weird.
Yeah, somebody stuck that R in there a long time ago just to be obnoxious.
Yeah, I know I'm supposed to say brewery, but I just screw it up all the time. I don't know why. I mean, I'm 68 years old. You'd think I'd have got it by now. Who knows?
Forget it. It's okay. You're forgiven.
From Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It's also a beautiful blue sky and sunny here with 32 degrees Fahrenheit. And happily, it's not nearly as windy as it was last night.
We had a little warm weather for a while, Brianne, right? And then all of a sudden winter's back. Yep.
Yes, it's very cold out there.
Yeah, it's not spring yet. We have a guest today from the University of Utah, Janet Iwasa. Welcome to TWiV.
Thanks so much. I don't know what the temperature here is. I feel like I came unprepared, but I also pronounced February wrong. So already two strikes against me.
I guess a lot of people don't pronounce it correctly. So Salt Lake City, we have you, and you know our friend Nels Eldy, right?
I do indeed.
So Nels Eldy's on TWiV, one of our other podcasts. And then Jason Shepard is on This Week in Neuroscience. You know Jason?
Yes. In fact, both of them are collaborators.
Wonderful.
Former, I think a current. Jason right now, we're working with him.
Nice.
And I've worked with Nels in the past.
Great. Well, we always talk about your work, so we've wanted to get you on for a while. You are a science animator, right? Is that what you call yourself?
I call myself a molecular animator just to make sure that people don't expect me to animate like
dinosaurs
or yeah, exactly. Or, the earth or something. So
okay. You animate molecules. That's great. And I heard you talk at a Carleton event which was, I guess, sponsored by your university. And I said, we have to get you here. So here we are. And let's talk about your background. I heard you give this story, and it was great. So start from the beginning, Janet.
Okay. Well, I guess I can share some slides then. So I will share my screen. So this is jumping a little bit into my career as a graduate student, but basically I got interested in animation when I was in grad school. I went to UCSF and I was in an actin biology lab. So my PI's name is Dyke Mullins. And we studied actin-based cell motility at the time. So one of the things that really got me interested in animation was actually seeing another animation. And so at UCSF, the Mullins lab, we had joint group meetings with Ron Vale's lab. So Ron Vale's lab studied microtubules and microtubule motor proteins. And they were right next door to us. And so we had joint group meetings. And, during these group meetings, people would, at the time, they were mostly talking about kinesin and how this motor protein walks along microtubules. And so this is an example of a figure that was shared during group meeting by a graduate student at the time who was talking about the mechanism of how kinesin walks. And so I saw like a ton of these types of figures over the years in watching Vale Lab presentations. And so you have these triangles representing the heads of kinesin, the motor were binding to the microtubule, which is represented as a line. And after seeing a lot of these, I was like, yeah, I get it. Kinesin walks head over head along a microtubule, just like that. And then what happened was that Ron Vale, so the lab, and especially some of the graduate students, they solved the structure, a partial structure of kinesin. And Ron Vale and Ron Milligan decided to hire an animator by the name of Graham Johnson to create this animated model of how they thought kinesin walks along microtubules. And a graduate student, again, showed this. So this is Sarah Rice, showed this during group meeting. And it just made my jaw drop because I think I had never totally appreciated kind of the dynamics and sort of like what the ATP, like what was really happening while kinesin was walking along. So this is an animation, just to describe it a little bit, I guess, for those who are just listening on the podcast, that you see kind of the binding and each individual step in an animated way. It really looks like something walking, some kind of creature walking along a sidewalk or something. But it really kind of brought things together for me in a way that I think none of these sorts of triangles and squares and arrows ever did. And so this was the first time I really started thinking, we have a lot of data. We have a lot of information that describes how molecules move, what their shapes are, and why aren't we using this to tell kind of more complete stories of how we think things work? Why are we just relying on circles and arrows and squares? Feel free to interrupt at any time. But, so this is kind of like the model figure that we would create in the lab. So where we would have like a circle, and this is actin, and we have a protein that binds to actin, and they come together and they form these structures at the leading edge of cells that allow cells to crawl forward. And so these are the types of things we did. And, and after seeing an animation, I thought we can do better than this. We can really start using animations to kind of construct a hypothesis, the story of how we think things happen. So at this point, I looked into whether I could take animation classes. So UCSF, it's a medical school. There are no undergraduates. There are no art classes. And so it took me a little while to figure out, like, how can I take an animation course, which is typically in sort of the art department. And I found out that I could take courses at San Francisco State University for free as a UCSF student. They had this sort of exchange program, and they had animation classes there. And so probably in my second or third year of grad school, I asked Dyke whether I could take an animation course, which was an art course at this other university, which was a really rather large time commitment. So it was in the spring semester, it was like all day on Friday for an entire semester. And I was really delighted that he agreed. And so what I would do is I'd go to this course, and I remember modeling like living room furniture. Like I modeled an entire living room set, like a upholstered sofa with a lamp and a table and like wallpaper. It was like Victorian kind of like living room. And then I would go back to lab, and I would try to figure out how to export proteins and import them into the animation software and start creating animations. And so this is one of my earliest animations, probably within a year of when I started creating them. And and this shows the process of polymerization of a protein known as PAR-M. It's a bacterial actin-like protein that undergoes dynamic instability. So it kind of builds and then kind of crashes. And so that's what this process shows. And, by the end of grad school, I was making illustrations like this. So this is the leading edge of the actin of the kind of a crawling cell showing all of the actin networks. And so this is the basis of, kind of illustrates the kind of work I did in grad school. And this is Dyke's
Janet.
wall. Oh, yes, please go ahead.
Can I ask you a question? So I have seen and looked at and enjoyed your animation for a really long time. And I'm sort of in awe listening to this because I would have guessed that you had a lot more of an art background. And so did you have any sort of additional art training before this? Was this the sort of your first endeavor into animation or into I don't know how much programming you had to do. Like, were you actually learning this as a graduate student, or did you had no other background in it?
So I think the last art class I had taken prior to taking this animation class was probably whatever was required in like middle school. And so there were, so I really had no interest in art. I remember there's like probably home ec or something. Like, I don't even remember. Maybe elementary school or middle school was the last time I had formally been taught something regarding art. I approached this from a very non-art background, I would say. And in terms of programming, a lot of the animation software has really been designed for artists to use. And so like most of the animators at Pixar and places like that are trained as artists. And so they don't typically, a lot of programming isn't actually required to do animation. That said, I think for molecular animation, it's pretty hard not to do any programming at all because, you have so many, proteins and I end up having to write some scripts to handle those.
I have a question about the assignment that was the living room furniture. Did you also animate that? I mean, did the couch move or no?
So the software that I learned when I was in grad school was a software called Lightwave, which is different from the animation software that I use now. And so that software, I actually never learned how to do real animation besides the one I showed you. This one I actually animated, I think, in Lightwave, but I basically stopped using Lightwave almost immediately and started learning other software because I found it really hard to use actually. But yeah, so I think I did animate maybe the light turning on, like nothing actually moving, but like the light turning on. And I made like the window shade translucent. So it kind of shone through and, it was a pretty, I spent a lot of time on that living room because I think I was just enjoying it so much. I just wanted to add another detail and, tufted pillows and and maybe it was also living on a grad school budget. I just, made the most ornate furniture that I could imagine. And so to kind of be sort of separate from my grad school life, my Spartan grad school life. So one of the things that, I think really made this career path possible for me was the fact that my graduate advisor, Dyke, was incredibly supportive of this kind of crazy thing that his graduate student was doing. Not only did he let me take the animation class, which is something like, I think at the time he wasn't tenured yet. He was, a relatively small lab. He let his graduate student go off and take an animation class. And then when I came back and I started making these animations, he was super supportive. And so after this course finished, he came up to me and he was like, if you want to keep doing animation, I think, you can spend your Fridays doing this, just as long as you want. So basically I had a day a week and I took him up on this offer, which was so generous. It would have never occurred to me to even ask for it because, that's basically unheard of to take, let a graduate student take a day a week to do something with kind of like, it wasn't clear like what the benefits to him were, but of course he did end up having tons of animations made by me of like basically everything the lab worked on at the time. I animated. And at the time, my skills were a little shaky. But he was very proud. And I think it really made it possible partially because everyone at UCSF knew about this, he would share my animations at a lot of his talks. And so people became aware that I was doing this kind of thing. And I think it really did open up my career path that I wasn't really hiding it. I was doing this as part of my lab activities, really.
So at the same time, you were still doing experiments at the bench, right?
Yes. So the other 4, 5, 6 days a week, I was doing my research. So by the end of grad school, I had this idea about animation. And this was really born out of my experience, like working with all of my lab mates, collaborating with them, trying to visualize their hypotheses, that animation kind of opened up these new ways of how we can synthesize biological data to kind of come up with this sort of visual hypothesis that we can share with people, with students, with kind of really anyone. And you can synthesize data like protein structure, the activity, dynamics, localizations. Like we can contextualize a specific reaction within like the context of a cell to really kind of tell that story. And so that's what I thought, this is something that's really needed, especially in research, to really kind of make sense of the vast amount of data that we now have. The next step was the hardest, I'd say, in my career, was figuring out what to do for my postdoc. I guess this was, that and the faculty position was also difficult. But so basically what I wanted to do was I wanted to get a postdoc where I could just do animation full time. And I had never heard of anyone being able to do this. But I had a real fixation on this. And I would, talk to my thesis committee, and they would be kind of like raising their eyebrows and shrugging their shoulders. And I would, email people I was basically wanting to try to find a fellowship. I wanted to find a way to get funded doing a postdoc, partially because I didn't think anyone would be willing to take a chance, spending their own startup or whatever on a postdoc who was doing something that was really out there. And so I was trying to find whether I could get funded by a fellowship. And so a friend of mine, Cynthia Furman, who is now interested, her career is in career development now. But at the time, she was a graduate student and she was interested in career development. And she had just had her finger on the pulse of all these different fellowships and things like that. And one day she told me about this fellowship that the NSF had called the Discovery Corps. And the Discovery Corps was under the chemistry umbrella, and they were trying to create new models for postdocs that would allow them to do outreach and education in addition to research. And so I wrote to the program officer and I called her and I basically said, I'd like to do animation. I guess I'd like to do animation that's like on the border of chemistry and biology. And I'd like to move to Boston, since that's where we are thinking. My husband at the time, my husband now, who was my boyfriend at the time, hadn't really decided what he wanted to do for his postdoc. But I figured if we go to Boston, he can figure out, there's a multitude of options. So I thought, Boston's a good landing spot. And I was like, does that sound like something I can do under this program? And she was like, absolutely, yes. They were actually looking for more. It was a pilot program. They wanted more applicants. And so she said, yes. And I have 3 people you might consider writing to. And so she told me these 3 people. I emailed them all lengthy. I did I read up on their research. I wrote them all lengthy emails about how I could animate the work they're doing. And I got one response, and that was from Jack Shostak at Mass General Hospital, Harvard Medical School. And basically to him, I said, I can animate the origins of life. I can animate how life formed on Earth, which is really what his research is right now really focused on. And so he wrote back and was like, I am really having trouble communicating some of these ideas to the really kind of like different audiences that he was speaking to. And so this sounds like a great idea. I'll support your application. And so basically I applied and I was awarded a fellowship. And this was really kind of a huge stepping stone that allowed me to kind of take the next step towards being a full-time animator. This is a protocell, an illustration of a protocell.
Yeah, I was wondering until you, of course, I didn't read. Actually, that's interesting. I was just looking at the picture. I didn't bother to read anything. That's the point of the animation, right? So that looks like a micelle with some sort of nucleic acid replicating inside, right?
Yeah. So the idea, is basically what did the earliest cell, that's something that we could call a cell on Earth, look like that gave rise to all life on Earth? And so one idea is that we had something called like a protocell that was basically a vesicle with replicating RNA in it. And so that's kind of like, how do we get to the point where we have a vesicle with replicating RNA in it is kind of the kind of a major sort of area of research is what kind of chemical reactions must have occurred, what kind of environments must have been on the early Earth in order for that to have happened. And so this is an illustration of what a protocell may have looked like. But, this is kind of the end point of like where we need to get to. So yeah, the communication part, as Jack emailed me, was kind of hard. And so there was a lot of interesting papers that came out of the lab, and this is one of them that came out of the lab while we were still there. It was kind of one of these early steps towards building this model protocell. And what's interesting about the origins of life field is that it's not just biologists, it's biologists and chemists and astronomers and geologists, people who all want to know, like, how did these early cells form and what might they have looked like and how might they form on other planets? How did they form on our own? And so you can imagine if you want to communicate to these diverse scientists as well as to a lot of the public who's also interested in understanding what the hypotheses are, it can be quite difficult to do that, especially if you have a publication like this, and if people are able to access it at all, then they have to kind of dig through these sorts of data and make sense of it. And so that was basically the goal, was to try and communicate the research and the hypotheses behind the origins of life to the public. And so as part of my postdoc, I went to this school. And so this is where I learned the software I know now. It's called Maya. It's considered the industry standard in Hollywood. And so most of the animation studios, many of them would use Maya as part of their production pipeline. And so I went to Hollywood to this school called Nomen, where I went, and you can tell it's not like a scientific school. We learned how to, animate explosions and like aliens. That's kind of their bread and butter. So I went there and I took these courses. I took a 10-week course that was like a crash course in Maya. And all the while, we're animating aliens and hands and things like that. And I was thinking, how can I use this to animate molecules? And so I'd ask like crazy questions, at least probably seemed crazy, where I'd say things like, say, and this is good because this is like a virology kind of thing. I'd be like, say you had a soccer ball and you wanted to take apart all of the hexagons and the pentagons and you wanted to lay them on the floor. And then you wanted to build it again from the bottom up. So just kind of all the hexagons and pentagons come flying in like that. And they'd look at me and they'd be like, why would you want to animate? Like, what kind of movie are you making? But then they'd answer, and I would have this sort of idea of like, okay, this is how I can, form a capsid or something like that. So I would just try to kind of think of like, what are real world examples of molecular things that I can kind of ask in a way that sort of makes sense. So I'd ask a lot of these kind of weird questions like that.
So Janet, at this time, what's the state of science animation? Is it pretty rudimentary? Outside of you? At
that time. Yeah. So this was over 10 years ago at this point. So at the time, there was maybe only 2 or 3 animators that I had met in the past, including Graham Johnson, who I mentioned before. Another one is Drew Barry, who's considered basically the father of our field. He did a number of really very high quality animations in collaboration with HHMI and Cold Spring Harbor on DNA replication, things like that. But so they were basically the only 2 people I knew, and I had emailed them both and asked them about their jobs and things like that. So there weren't a ton of examples, and there definitely wasn't really a field. There were like a few people. And another person, David Goodsell. So David Goodsell at the time was already creating beautiful watercolors of these molecular scenes. And so David was also someone I reached out to and asked him about his career and how he got to where he was.
But Goodsell was at Scripps, is that right?
Yep.
And the other 2 individuals, were they at academic institutions as well?
Graham Johnson at the time was freelancing as an illustrator. So he had started working on a textbook that is authored by Pollard et al. So kind of a cell biology textbook with Tom Pollard and a number of other authors. But he had met Tom, I think, through this kind of KINESIN connection. So he was already working on more cell biology type things at that point, and was living in Boulder. and taking classes at UC Boulder. So Graham went on to get a PhD at Scripps, working with Art Olson and David Goodsell.
Okay.
Yeah, so just to, some of my early animations look like this. I don't know if you can hear this. It's actually playing noise into my headphones. But this one is showing, can you hear the narration? I'm guessing not.
No,
okay.
No.
So this is showing a vesicle in cross-section where nucleotides are able to enter. So this particular vesicle is made out of fatty acids, which are able to flip-flop between the inner and outer leaflets. And it was thought that basically hydrophilic kind of things like nucleotides would be able to enter the vesicle that way. And with a high enough concentration, you can then start polymerizing RNA. So this is one kind of idea that was based on experimental data that was taken in the Shostak lab.
So that modeling is with they're not lipids, they're fatty acids.
These are fatty acids, yes.
Okay, and so the permeability to a charged molecule is going to be different for something just made of fatty acids as opposed to lipids. Is that right?
Yeah, because the fatty acids can just flip-flop. They can just kind of turn in. So that's some molecular dynamics had kind of shown that as well. So they're much more flexible. Whereas phospholipids, you need special proteins basically to flip them from one side of the membrane to the other. Whereas for fatty acids, that's not the case. And so we had animations where we built vesicles from micelles, and that would require flip-flopping. So basically to keep an even kind of density of these fatty acids on both leaflets.
So I presume you can demonstrate experimentally that this can happen?
They did. In fact, this was based on experimental results.
Okay.
Yeah. So one of the things in terms of the outreach, so I made a lot of animations. Some of them were kind of basically like, if you could look in the test tube, what would it look like? Like nanoscale, that was an example of one that was like a nanoscale type of animation that visualized experimental results. A lot of other ones were a lot more hypothetical. So they are animations of like, how do fatty acids form on the surface of minerals, like a mineral face. And all of these animations, I basically put on this website that was called exploringorigins.org. And this was considered like an online exhibit. And the other part of the project, which I should have mentioned earlier, is that this project was also co-sponsored by the Museum of Science in Boston. So one of the things that I was really fortunate to have gotten the opportunity to do while I was in grad school was to go to meetings on visualization, on science visualization. And at some of these meetings, I met people from museums who were interested in various aspects of communicating to the public and visualizing science. And so during these meetings, I met some people from the Museum of Science in Boston, which is like 10 minutes away from the Shostak lab. It's very, very close. And so I was very fortunate to basically convince some of the people at the Museum of Science that I should put together an exhibit on the origins of life as a product of my postdoc. And another thing that's worth noting is that this was a chemistry postdoc, so it was only funded for 2 years. And so I had this like 2-year period to like learn animation software, create a bunch of animations, put together an exhibit. And it's kind of a small miracle that all of this actually happened. So here's the picture of my exhibit at the Museum of Science. It was on the museum floor for 5 years. In addition to having this kind of like touchscreen monitor that played all these animations and explained things, I also gave a bunch of presentations at the museum. So just live audience, kind of whoever was walking around, I would give basically a 15-minute, I think, presentation about the origins of life.
And you did the whole exhibit yourself? You made every aspect of it?
I had so they had someone who helped me with the narration. So the narration, there's someone who was considered the voice of the museum who narrated everything that there was kind of on the floor. And so she recorded all of the narration. But other than that, like even the touch screen thing, I coded that. So one of the nice things that they did was when I was giving my live presentations, they had a lot of helpful feedback on how to make this the most public friendly. So they gave me a cap on the number of jargony words I could use that I had to define, like vesicle. Like I couldn't get around not saying vesicle because they were like, maybe you can say balloons. And I was like, no, or bubble, and I was like, no, I think this is one of the words I have to define. So things like that, they helped me kind of figure out like how best to communicate those kind of concepts to the public.
Yeah, we should have that here on TWiV. We should have words that we're not allowed to use. Boy.
Yeah, forget it. That'll never work.
I think it's just you have to know to define them and define them using terminology that is like common terminology. So it's helpful to know that. And so I always do think about that, especially if I'm giving more public talks is, you could give more analogies or things like that to kind of get the idea across. And the wonderful thing about animations is often you don't have to talk about it that much. You can just show and not have to, name all the proteins or something. So often I'll be like, the red one does this or something,
How do you do vesicle? How do you define vesicle?
I just used, I had that as one of my terms I use. So I defined vesicle. I think I probably said it was like a bubble, like a bubble, but I didn't call it a bubble.
A bubble in the rest of your talk. Okay, got it.
Yeah.
It's so hard because, I know what a fatty acid is. I know what a lipid is. I know what its properties are. Vesicles make sense to me. And to come in cold turkey and try and understand all that stuff, even understanding what hydrophobic and hydrophilic or polar and nonpolar are is a huge challenge for somebody who never thinks about this stuff. So to describe it is tough.
Yeah. Yeah, I think that's true. And it's also just hard sometimes to put yourself in somebody else's shoes and just imagine what would be the most straightforward explanation.
Well, and I suppose that's where the animation helps, because you can have pictures to show it. So do you get, did you have an opportunity in doing the museum exhibit to get feedback that helped you understand how well you were communicating what you were trying to communicate? And I would think that would be a learning experience. You would learn, oh, okay, I got to do this.
One of the things that was really interesting about working with the museum is that they do a lot of assessment. And so this is something that I am not I don't do as often as I should. But one of the things that they had me do is they had me ask people about their assumptions about evolution and the earth. Because this was one of my goals was to try and explain how evolution happens on a molecular scale. And so therefore, how does that kind of explain how it happens on this sort of larger scale? And with protocells, it's pretty easy because all you have to do is like a 1-point mutation in the RNA and it may no longer have that function, and it may even have a new function. And so like describing that as a step in this evolution was sort of important to me. But, so they had me, have like a little booth and then people would come in and I'd talk to them basically about evolution or, say like, at what point did these different animals or different organisms appear on the earth? And so you get a feeling for what people's background is, what kind of things that they have preconceptions about. And so those kind of things, if you know that, then you kind of come in with a more, what you're kind of, you should talk about or what background you should provide. So I think knowing your audience is really crucial. But I think, that requires quite a bit of research. And then there's assessment on the other end of how effective was it? Like how much do people actually remember afterwards? And so we did assessment on both sides of that, both kind of before I put together the exhibit and then afterwards.
Cool.
Yeah. So, one of the things that really stuck with me was when I went to the museum and I gave these live presentations. And I have to say this kind of bothered me at the time, but, I had more than one experience where I'd give this presentation on the origins of life. And then somebody would come up to me and they'd ask something that was like totally unrelated, right? They'd be like, so how did you get into science or something, and at the time I was like, oh, that's not really what I was talking about. I don't want to talk about myself. I want to talk about protocells or whatever. But you kind of realize that these science museums really offer, especially when you have scientists coming in and giving presentations, an opportunity for people to come and really kind of interact with scientists in a way that is kind of hard to do, especially in pre-Twitter, kind of days, that it was really not something, people didn't really have those opportunities. So, and people would bring me, push their kids up to me and be like, ask her a question, like my daughter's interested in science. And and I think afterwards, at the time I was like, ask me something about what I'm doing research in. But now I really appreciate, I think back on that and think about, like how kind of awesome that was.
I think the personal parts are important, right? It makes you more than, it makes a scientist real, right? You're a scientist, you're also a person. It's important.
Yeah. And I think part of it was like, basically, why are you interested in this? Like, what makes you tick? How can I get my kids interested in this to the level that you're kind of obsessed with this, kind of thing?
When people ask those kinds of questions, that's coming from somewhere where they're asking, they're asking for themselves. It's interesting. I look back on my own life and I can remember wondering if I was just saying I was interested in this or if I was really interested in it and where it might go. And I look back on all that and I realized that, I was smarter about myself than I thought I was. And I'm glad I followed my instincts. And these people are, among other things, probably asking you, are my instincts right here?
Or even just like, how does one get into this? Like, what do you do to become a scientist?
Right. Well, I think it's also sort of a very humanizing thing in helping you realize that this ideal that you're imagining is something that you can actually do. And you can think about the steps in between you and there. I mean, to be honest, that's part of why I asked you about the art background is as because I was looking at this and I was like, man, maybe there's still hope for my poor drawing skills.
I think it really does help to kind of understand how people got there and being able to have that opportunity. And I think it's one of the things that I really appreciate about Twitter now is that there are a lot of people who are trying to demystify what doing science and being a scientist means so that more people, to kind of open the doors, hopefully, for more people. One of the things that happened out of this project was that the project got written up in the press. It got used. And so I got asked this once is, what makes a visualization good? And I basically said, a good visualization is one that's used. Like if it's not used, then it's not a good visualization because nobody's using it, right? So it's basically about utility. And so one of the things that was really great, and this is due mainly to the fame of Jack, is that it got used, it got used in Wired, it got used in the New York Times. And I think it gave this kind of visual handle to understand these hypotheses on how these protocells formed and what the importance of them are. So, after this, basically I had to figure out my next big leap was going from my postdoc to whatever the next thing was. And so I was really fortunate. The timing at the time, I had a 2-year postdoc. My husband is a neuroscientist. He was doing a kind of more typical 5-year postdoc. And so I had some years that I had to spend kind of doing something in Boston. And so I really lucked out in getting a position at Harvard Medical School in the cell biology department as a faculty doing animation. And so this was kind of also a dream position. And so basically during those years, I worked with a number of different people animating all sorts of cell biology. And so this is an example back from 2012 of CLATHRIN mediated endocytosis. This was done with Tom Kirkhausen. And so basically this is a pretty fast version of this animation. It basically shows the formation of a vesicle, an endosome, and then kind of what CLATHRIN is doing to do that. I have a much slower version online, but this, so the way I think about my work, and this is my last slide, is if we rely on illustrations like this, which is a more typical 2D kind of illustrator type PowerPoint drawing of endocytosis. If we rely on this, we're potentially not, there are a lot of things that could be gained from doing 3D animation. I think what I found is that there are a number of insights that researchers can get when you're able to kind of really visualize things, the kind of the movie that plays in your head. That's the way I think of it, is that for anybody who's been doing research for years, decades, they had this movie in their heads of how things happen. And this is based on not only published data, existing ideas, but also a lot of things that they aren't really sure that they're happening, right? There are things that they're actively being testing. But we can visualize this hypothesis, this kind of the movie that plays in their head. And if we can do that, then people are able to ask better questions about how to and design better experiments. So that's kind of the idea, is if we just stick to these 2D type things, simplified illustrations, we're missing out on a lot of things. And the other part is about science communication. So, like one of the things that I say sometimes is, for CLATHRIN mediated endocytosis, if I talk about that and I show you this figure, and I'm like, yeah, there are many labs in the world, people spending their entire lives understanding this process, and I show you this drawing, it makes people kind of like non-scientists be like, really? They're spending their entire lives studying this like kind of like all these circles and stuff. Looks pretty simple. But then you show the 3D animation that shows all of the dynamics and the complexity and the crowdedness. And they can understand why this is really fascinating and interesting. They can come up with questions, kind of like what happens here, what happens there. Children can ask really great questions about endocytosis if you show them really what you think is happening. And so I think that's part of it, is that I think, these animations really humanize molecular biology by showing why molecular biologists are so interested in these things.
So you spent a couple of years at Harvard, I presume. I mean, what did you... You're an independent person, right? So you could do whatever you want. So how did you pick what you wanted to animate?
A lot of it was by who was around, the collaborators. So all of my projects now are in collaboration with researchers. And so since leaving Acton, I haven't animated something that I personally worked on. And so I rely pretty heavily on collaborators to kind of tell us what they think is happening. Tell me about this movie playing in your head so that I can animate it. And so that largely has been people approaching me and just saying, I have this project. I think it would be great to animate it. I've had trouble communicating it. And that's the major way I get collaborators.
So how did you get the job at Utah?
So Utah, my husband, I'm a spousal hire. So my husband, like I said, is a neurobiologist. He, one of the places he got an offer was from Utah. And so we came here. I came here on his second visit, and, I gave them my spiel, my talk. And the biochemistry department was clearly a good fit for me and my interests.
So clearly this is an untraditional hire. Okay. So typically in a science department, your promotion and tenure are based on teaching, service, research, and research, among other things, is defined as, or is evaluated in part at least, on bringing in money. Okay. So do you have ways to fill those slots?
I do.
or are they just trashing all the rules and saying, do it?
No, I still have to fill out the same forms as everybody else. So, we're at a medical school. So in terms of teaching, I participate in medical school teaching, pretty much the same as the other faculty in my department. And I also, in addition to that, we also collaborate with faculty who are primarily in education and create animations with them. So we have one teaching grant right now to animate action potentials and their kind of their role, both at the molecular level and sort of at a larger level. So we do things like that. And I've also participated in textbook illustration. So kind of education at different levels. For the research, so we have been, one of the things that the department did change or ask, so basically when I was I was promoted to a tenure track position. I was asked to look over the requirements for tenure under my department and to see whether anything needed to be changed. And so one of the things that I changed was the kind of the contribution. So does it have to be a high profile publication? I changed that to contribution, kind of a broader definition of what a contribution could be, because, like a lot of our animations, we don't publish them in like journals or something, but they are something that are widely seen and contribute to kind of our understanding of scientific knowledge. And so I think by broadening that, that kind of helped encompass the things that I plan on doing, as well as hopefully other people in the future who may contribute to the research and kind of the research sphere in different ways.
Well, good for you and good for Utah.
Yeah.
I'm super grateful. My department and the University of Utah in general has been just kind of the most welcoming and kind of warm, kind of academic home. So it seems
really to me that at every stage you've sort of broken new ground. That's really, and it's really important ground to break, because the result is phenomenal. To actually be able to visualize this stuff is really an important contribution. It's terrific.
I want to know a little about trainees in your lab. So tell us about that.
Yeah. So right now my lab is I have 4 postdocs and an animator. So there are 3 postdocs. Yes. Anyway, we have 4 people in the lab, 3 postdocs and an animator. And basically, one of the things I also decided was, for me in grad school, having my own research project, my kind of bench that I was doing at the bench, was a kind of a major, obviously the major thing I contributed to science in grad school. And so for me, the way I think about animation is this is a technique, that I think would make sense for postdocs to learn as sort of like their kind of something that they would like to apply in the next part of their career. And for graduate students, if they already have a research lab. So basically, I would accept graduate students only as a co-mentor, where they are essentially animating the work that they're already doing. So they've joined a lab, they've gotten some results, they'd like to animate these results. Then they would be able to come to the lab, train in animation, and be able to create those visualizations. But we don't take graduate students, without another mentor, a primary mentor.
And so, have your trainees gone on to do animation types of careers?
So I only have, our lab has only been in existence for 2 years.
Oh, okay.
And so it hasn't been very long. I've talked a lot to our postdocs about, what they'd like their next steps to be and what that might look like. And given all sorts of advice on how to get there. And I'm very confident that they'll be able to find something that's a good fit for them, because I think there is just a lot of interest right now in trying to visualize really diverse molecular mechanisms. And I think people are becoming more aware of this as kind of a field.
Yeah.
I'm interested. Go ahead.
I think you're training the next generation of molecular animators, right? That's probably what you want to do.
Yeah, and I should mention that there is a field of medical illustrators who also overlap in terms of doing molecular visualizations, and so that's a master's degree program. And actually, many people in my lab have master's degrees in medical illustration, in addition to PhDs, and so. I think there's already a kind of a field there, but I think where, what sets our lab apart a bit more is this sort of interest in understanding where visualizations can help research, where we can kind of push our knowledge and kind of understand better how things work and feed that back to researchers. So I think that's kind of a bit of a difference in terms of who we think are kind of our main audience is. And I think for a lot of our projects, it really is the research community, and hopefully allowing them to communicate their ideas, not only within the research community, but outside of it. But really our audience is mainly towards researchers.
And
I think that
Go ahead, Brian.
I think that not only are you training the next generation of molecular animators, but I know that myself and a lot of people who are in teaching focused positions use your animations all the time. And I find them to be one of the most useful things to show my students to really help them understand both, the general process of endocytosis or whatever I want to tell them, but also just the ideas of biochemistry and that fact that there is dynamics in these molecules. This is sort of helping general training in so many ways. And I can't thank you enough for all of the ways you've helped.
Thank you. So one of the things I guess I wanted to touch on a little bit that leads me into like the coronavirus project that I definitely wanted to share with you is, one of the things when it comes to teaching and when it comes to public outreach that I've fielded a lot of concerned questions about is, you have this one animation that shows this one process and everyone thinks that this is the way it happens or whatever. You show it and like it's not clear where the data comes from. Is this a consensus model? Does the entire community really agree on this? And what I would argue is at the molecular scale, there is no consensus. It's very, very difficult to have a consensus on something we can't see, right? So I think it gets harder. It gets hard to do that, and communicating that is very difficult. And so, if an animation ends up being incorrect in one or two ways, like how can you change that? Students will see it and they might get the wrong impression. So I think this is something that's a bit difficult when it comes to animation is the lack of transparency, because there is, parts of the animation are from like pretty hard data. We can cite a paper and this is exactly what it looks like. And then there are parts, especially because we're showing dynamics, right? How things move, that are very gray areas where, there is maybe no consensus. Maybe they're competing hypotheses. Maybe, there's just like one conjectural model that's out there that's on shaky ground, So I think, how do you show that in animation? Everything kind of looks solid and real. So that's been an area that I think has been something that I'm very interested in thinking about, partially because I think it's really important, because we are trying to show hypotheses, and hypotheses are, they get edited over time. They get edited by the community. They, like something might bubble up to the top as kind of the favorite hypothesis, where I've animated, something that may not have been. So, so I think that's been something that I think about a lot is how do we build consensus as a community, and how do we visualize that?
I like Brianne, I use animations of all sorts in my teaching. I find the students get so engaged when they see a short movie, right? There's just nothing like it. And you don't have to talk. You just show it. But you can find errors or things that don't look right in all of them. And, I like to point them out, just like there are errors in movies, right? But that's fine. I think that's part of the learning process. And you can revise, as you say, you can revise them as you go on. I think that's fine.
Yeah. I think, and that's important to do, is kind of keeping track of that too. So, I have this coronavirus SARS-CoV-2 animation that shows entry. Going into it, this is what I was thinking about. We have so many papers that I'm sure you've discussed many of them about SARS-CoV-2 that have been published by many labs, some of which have been studying coronaviruses or their ilk. For my idea that I proposed to the NSF was that one of the ideas was like, can we use visualization to show, what we think is happening, and then to basically allow people to comment, like this part, for us to annotate. So this is where the data came from. These are the structures we used. This is the references for how this binds to that. And also to be able to allow people to ask questions like, for example, for entry, does endocytosis or, does entry only happen at the plasma membrane, or does it happen in an endosome? So this is actually a question that came up multiple times that, made me think, maybe we should revise the animation to show endocytosis in addition to entry at the plasma membrane. But at any rate, so that's the kind of thing I was thinking about, is if we have more comments at a specific part of the life cycle, then we know that there may be a lack of consensus there. If there are a lot of people saying, maybe this is an alternative thing, and actually this is the way it happens, and everyone's giving it a thumbs up, then we need to reanimate that section to show that. So this project is about animating the life cycle, the entire life cycle, not just entry. And so we're about right now we've only released entry, and we are working on the other sections that we're releasing later this year. And we have it all embedded in this software that allows people to see the annotations and also be able to make comments.
Would it be possible for you to show it to us now, the entry part?
Yes. I'm just going to show the animation first and talk through it. So this is kind of the beginning part I consider to be like the anatomy. So basically just showing the different parts of the virus and where they are relative to each other. So we have the viral genome inside. Of course, we have the spike proteins on the surface, and they're waving around. And so now we're basically approaching an epithelial cell in the respiratory tract. And so on the surface of the cell here, we have ACE2 proteins. They're shown in purple. So ACE2 is able to bind to spike. One of the things that's a little subtle here is that, the spike is in the upstate when it binds to ACE2. So that's shown in the animation, but maybe not super clearly. We have all these glycosylations on the spike proteins that are kind of waving around. Now we have TMPRSS2, the protease, coming in and cleaving the spike protein in these specific sites. And exactly what happens to these cleaved proteins afterwards is not entirely clear. We have them kind of sticking on to ACE, like remaining on ACE2 and then kind of like dissipating away. And then, the spike protein undergoes this major conformational change where it basically injects itself into the membrane of the host cell and then goes through another conformational change that forces the fusion of the membranes. And that allows the nucleocapsid, the genome, to enter the cell. So that's what the animation looks like. I also wanted to show you what the animation looks like embedded in our annotation software. And so using this software, basically you can hover over different parts of the protein and you can see basically the annotation. So the annotations are shown here on the right side. So if we click on one of these proteins, then you can see all of the annotations associated with spike. And here on the other side, so down here, we have all of these comments. So each of these little lines represents a comment. Most of these actually were made by me, where I was basically trying to take Twitter. The people mostly commented on Twitter, despite me trying to get them to go to this website. And so I basically kind of commented on the different things that people asked about or commented might not be correct. And like I mentioned, the thing that I think more people mention than anything else is the idea that the point of entry might not only be at the plasma membrane. And so there were several people who asked about that, and that I added here. So, so I think for entry, it might be, kind of the most relatively well understood part of the life cycle of the virus. And so we're really interested in seeing what happens next. So we're showing transcription, translation, the formation of double membrane vesicles, and egress. And so all of these, I think there are a lot more kind of open questions about what's happening inside the cell. So I'm hoping we get a lot more kind of activity and questions and sort of like competing ideas. So one of the things that I really want are people to disagree with what we've shown, just to get an idea of like what, are we far from a consensus here? Are we getting closer? And for the second part, so for transcription, we do already have two competing hypotheses that we're going to animate. So this is based on conversations with a number of people who published relatively early so we basically got in touch with people mostly last year in the spring and summer of 2020 and interviewed them basically on the mechanisms. And we're currently animating those mechanisms now based on kind of these conversations and their publications.
So thinking specifically about the entry video that you just showed, but beyond that as well. In the entry video, how many of those proteins that are pointed out do you actually have atomic structures for?
For the spike proteins or?
No, well, beyond, I know you got an atomic structure for the spike protein. And
for all the other ones.
But TMPRSS2, nucleocapsid, the other membrane proteins, do you have atomic structures for those?
So we do have partial. So, in general, I depict flexible domains. So those generally we don't have structures for, as I approximate their length. So I look up how long is this region between two structured domains where we do have the structures, and then basically say, okay, this is going to be a flexible linker that's going to allow this part to go kind of waving around. So those we don't have structures for. So for example, the flexible regions of Temperis 2, there was no structure for that, but I approximated it based on the length. The structured domains if there is no published experimentally derived structure, then I took whatever was predicted. So I have for some of the structures predicted structures that I use, and I also included in the annotation the links to those. I think the hardest one here was the conformational changes of spike. So Jason McClellan kindly gave me a structure that allowed me to make this animated version of spike undergoing this conformational change. And I think those dynamics are really the hardest things to capture because we often don't have experimental data for all the states. And so it requires some, modeling in order to really show those.
Yeah, what do you have? You've got pre-fusion and post-fusion for sure, but the way it's illustrated, there's a sort of an intermediate as well, where the fusion peptide has penetrated, but that second conformational change that you showed.
That was Jason McClellan gave that to me. So he modeled it, and I saw him give a talk, and I was like, oh my gosh, we need that structure. And so I emailed him right afterwards, and he sent me the structure. He's acknowledged on the animation, and he also gave us a ton of feedback. So this is actually the second version of Entry, and so he gave us a lot of feedback on the first version that I made changes to. So this is actually the second version that I'm going to make some additional changes to potentially.
So that intermediate structure is a model. It's hypothetical, right?
It's hypothetical.
Okay, because that's going to be, as I know this is where you're going, but I really like the annotation because being able to hover over a given structure and understand, do we really know this?
Yes.
Or is this what we think? Because that always, when I was learning this stuff, that always confused me because I would see something that, in the old days before we had almost any atomic structures, there'd be this blob, okay, and a couple of side chains in a cleft in the blob that was supposed to be the active side of the enzyme. And I thought, oh, they really know something. But no, all they knew was that there was some chemistry that said if you wipe out a sulfhydryl, the reaction doesn't happen. Okay.
Yeah, no, I think it's important to give that. I think it's actually hard to provide all the nuance within even the annotation because, you don't want to write a book. So I think it's kind of a first step towards that. And, maybe what becomes sort of interesting is how do we see the evolution of a hypothesis over time where, we're limited by the data we have and kind of what was kind of considered to be the consensus in the beginning. And as we move further in time, we have more specific ideas of how things work. And, some things are shown not to be correct and these things evolve. And so I think showing that evolution and what's changed and, is really kind of interesting and is the way science moves forward. And so this is something that I would love to be able to kind of do in a long-term project.
This may be more confusing than helpful, but I wonder if it would be worthwhile to, in any given molecule, use colors to indicate this is what we know and this is what we hypothesize.
We had a workshop with a bunch of molecular animators to talk about how to visualize uncertainty. And it's something that we're like, can we use transparency and like things that are less certain are like more transparent? And I think we're like, can it just be a layer? Because you don't want to take away from the understandability of an animation by adding all we'll make it fuzzy or something. And so I think that would be super distracting if you're watching an animation, all of a sudden something really fuzzy comes in, you can't even tell what it is. So ideally you would see the animation in full clarity and then you'd have like some uncertainty filter that you'd add on and then all of a sudden, but that's very subjective because some people, right? I mean, it's how do you quantify how close we are to knowing certainly that this happens this way. And so I think that's the thing that's a little difficult, because of course, some people are going to believe very strongly that they're right about this specific hypothesis. And you don't know whether they're a rogue, rogue scientist or something, or really kind of like maybe everyone actually thinks that, even though, there hasn't been anything published on it yet, or whatever. So I think it's sometimes it's very hard. It's very hard to tell if you're, especially I think without meetings to go to, a lot of these discussions happen in the Q&A after a talk, and you get this idea, okay, a lot of people don't agree with this idea, especially if you're an outsider like me looking in. It's a little hard to figure out where disagreements really are happening in the field.
So I really liked how in the animation that you showed us, it immediately gave an idea of a question that somebody could study of what happens to those cleaved pieces. I think it just makes it really obvious of, okay, that's an experiment somebody could actually try to figure out.
In Twitter, somebody asked about it because they're like, could that be like, a target for an antibody or something, all these like cleaved pieces of junk. So and I was just like, I think one person was like, they probably stay stuck to ACE2, but I think that was really the extent of which we got with that. So it's definitely an open question. I think to them it looked very unnatural. So they're like, is that really what happens? And I was like, basically, I don't know. I don't know if we have much data telling us one thing or another.
Yeah. So do you have any projects or kind of long-term ideas about iterating some of the other animations you've done with updated new data? I was thinking about the HIV one that you've done and some of the cool data that's come out recently on that.
Yeah, so we have a project called the Science of HIV. So this project is being funded by a CHEETAH center, so a P50 center that's funded by NIAID. And so for that, basically, I've been working on this project for many years, ever since I moved to Utah. And so I've animated the entire life cycle of HIV. And we have been editing it. So there are a bunch of parts that I put in, I'd say low resolution in the first version, RT, reverse transcriptase, is really hand wavy, just kind of, and so some of the things that we have to change, there's a lot of new information about where that the entire capsid goes into the nucleus. And so, that's a major thing. We're redoing that part now. So the entire capsid's going to go into the nucleus, and RT is, and so RT is kind of working on things, but not completely until it gets in. So I think there's a bunch of things that have changed, and so that's been really fun to work on. Escorts during egress, there's been a lot of different kind of models hypothesized on how budding works. And so that's been a really, really nice way, because we're both involved in sort of the research side of things, working pretty closely with our collaborators within the center, as well as trying to keep track of how the field is changing. And this is definitely a major way where exactly does RNA become accessible has been a major kind of storyline change in the animation since we started.
I think it's clear that you need to have a science background to do this, right?
I mean, there are a lot of animators that may not, and I think it kind of depends on their ability to kind of dig through scientific literature, and so I'd say, people ask me, was a PhD really necessary to do what you do? And I'd say yes, in many ways, especially because I'm a professor. And so I think there was some requirements there. However, I think there are a lot of people who do molecular animation who are quite knowledgeable and are able to kind of really dig into the literature. I think some of the nuances can be a little harder. But yeah, I'm a little hesitant to answer absolutely yes, you need to have a science background, because I think there's definitely good work being made by people without PhDs.
It seems to me that it's a little bit like a science writer also. It depends on how well you can communicate with the scientists or the science in the literature. If you can do that well, and then figure out, and you're willing to, get it vetted, and say, did I get this right?
I think, and that's true for, a lot of the animations we create. the first draft is just wrong. Like we had this conversation, we go back and forth, you've given me figures, and then I create the first animation, and there's always something that's wrong because I've interpreted something wrong or like whatever. Like there's only so much you can get back and forth through just like verbal communication. So I think no matter what, like getting the picture right is always going to take iterations and just the ability to understand, well, like you can't get upset because, like, or people come up with new ideas. They're like, oh, that made me realize something. And that's, for me, as with someone with a research background, that gets me excited. But, like if you're in a studio, you have a timeline, that can be really annoying, right? Like they're just kind of change their mind about how the hypothesis works. And like, you can't, like, I think that makes it harder for certain, depending on like what your work situation is. But for us, I think it's often really exciting to think about how visualization is playing a role in making people think differently about how things work.
So, Janet, is this, given the past year, is this the kind of work you could easily do at home?
I have been doing it at home, or in the outside in the Utah wilderness. Our lab has transitioned very successfully, I'd say, to working from home. Really pretty seamless, I would say. Yeah.
So you just need a computer, right? Basically, and internet, I suppose.
Yeah. Yes. Yeah. So everyone is working from home and has been doing so. And I'd say our production wise, things really haven't changed. I think one thing that might change is how people feel about going back to work. Like I think there are members of the lab who say, I feel so much more productive at home because I have no distractions. And so I think I would like to, spend at least a couple days a week working from home, which is also, doable given kind of the way we work.
I think you touched on this, but is there no such thing as a scientific illustrator or animators conference?
There are visualization conferences of different types. So the medical illustrators have a conference that's run by the American Medical, the AMI is the acronym. And so they have a conference that they have for kind of people who are accredited medical illustrators. They're also more within the sort of the research side. There are a number of visualization centric meetings. So I regularly attend a Gordon Research Conference on visualization and education. There's also a meeting called VISB that happens every other year, that is also specifically on visualization of biological data. So that includes, kind of more data visualization in terms like, visualizing genomes and sequences and things like that, in addition to like 3D structures and animations.
Do you ever get asked by Hollywood to help with their movies?
Why haven't I? I have not. I mean, so, I have gotten asked to look at, museum exhibits. But yeah, I think I have been asked to make animations that I think are questionable. And I basically say no, that it's kind of not within our wheelhouse to be overly hand wavy. And I have also been asked to do pseudoscience, which was a hard no. And really I worry that in that case, if someone was asked, a medical illustrator or someone with maybe less of a science background was asked to do that, they might do it without realizing that this is total bunk. So I think, that kind of thing I've also encountered, and it makes me quite concerned. But yeah, so no, I guess if I was asked to do something that would take me away from being able to be accurate, I would just say it's not within kind of the scope of what we do.
Janet, we talked about your SARS project and the HIV project, but one of the ones that caught my eye on your projects page is the Seeing Diversity project. And I thought maybe you could just tell a little about that.
Yeah. So last year during Shutdown STEM in June, the lab, basically we had several discussions on Slack about reading more about racism in the US and how it affects science, our scientific fields. And people really wanted to think about how we could do something. And so, kind of out of discussions, what we talked about, and this also came out of this really interesting experience I had giving a talk where a student, a very, I would say, very brave student to ask this particular question, basically was like, so you work with a bunch of famous people who are very established. What about everybody else? You're helping these very established people basically kind of like promote their own ideas even better than they already have, and they're already established. What are you doing for everybody else? And I was like speechless. I was like, I don't know what to say. And, what I did say was like, I will work with anyone, right? So like, if it's kind of within our wheelhouse and I will help people find funding in order to help them, work with us. I have written, different proposals and things to, so it's not like just HHMI labs or something. However, I felt really bad because there is this element that like, yeah, like what about everybody else? And so anyway, I was thinking about this, like, how can we help promote, and celebrate the diversity that we have in the sciences? How can we give back? And so that was kind of where this idea was born for Seeing Diversity. Is basically one person from the lab every month volunteers their time to work with someone that they admire, and they will reach out to them. So we have kind of a list of people, kind of a diverse scientists. So this year we've really been focusing on working with Black scientists, and basically, asking them, how can we help you? How can we illustrate something that can help you communicate your science to diverse audiences? Use it on your website, on a cover illustration, your talks, and a lot of them are still relatively young. So they're trainees, they're graduate students, postdocs, and young faculty. And so that's what we've been doing every month is basically creating this illustration and then posting it on Twitter. So we post a tweet that shows the illustration that the lab member made and something about the scientist, maybe something personal, definitely something about their science and their contribution. So yeah, we're actively accepting anybody who wants to nominate themselves or somebody else. We have a list that we're just kind of keeping a list of people, and every month somebody from the lab goes through and just selects someone. And right now we don't really have so they've been 3D illustrations, 2D figures. We don't really do animations because that's a little too time consuming for us to do on a pro bono basis. But yeah, so that's what we've been doing, and it's been really fun. Like, people love it. It's a way to kind of be more creative, to meet new people. So it's been just a great way to kind of make new collaborations and kind of diversify kind of the science we know and the people we know.
Yeah, and the illustrations are really great. I mean, it's just fun to look through them all. Yeah.
Well, I think we have kept you enough, Janet. This has been terrific. So thank you so much for joining us, Janet Iwasa, University of Utah.
It's been a pleasure. And yeah, so the only last thing I'd like to leave with is, yeah, please do. I know, listeners to this podcast will be interested in, hopefully interested in our SARS-CoV-2 animations. So keep an eye out we have entry, and we will have the rest of it. And we would love to get more comments. And so that's something I would love to plug as something that we would actively love to see more comments from. Doesn't have to be from a scientist. If you have questions, or you don't understand how something works in the animations, we'd love to hear from you.
Yeah, we'll put links to your animation lab in the show notes so people can find it.
Great. Thank you so much. It was a real pleasure.
Thanks, Janet. Pleasure. Take care.
Thanks, Janet. This is great.
All right.
Cool.
Bye.
Bye.
I also went down the hole of looking at the Cryo EM 101
Me too.
animations. Those are cool.
Yeah.
So yeah, it's all great stuff.
So much of her stuff is just so amazing.
It's very good stuff. Yeah. Great for teaching.
Mm-hmm.
Well, it's great. She's created her own career.
Yeah.
Good for her.
That's what I think is unique. She has it created, and the university created a position for this. It's not a traditional, which I think is very good of them. It's a wise thing. And, not every university would do that because they have the myopic grant-driven, requirements all the time. All right. I thought we'd follow up by a brief discussion of the latest vaccine news. That is Johnson & Johnson Janssen's Ad26 COVID-2S results. You know, the FDA went over them on Friday, and they look really interesting. So I will put a link to all the documents. They're just like the other two that we've gone over, Pfizer and Moderna. There's a page with multiple documents. You know, they include a big one with the phase 3 data. There's actually a slide presentation as well by 3 different people from the company. There's a cool presentation about the what is it called? VSafe data, you know, when they ask you, it's time for your daily VSafe after your vaccine. So we have numbers. So check those out. They're really interesting. However,
the slideshow is great.
Slideshow is very good.
Very good.
Very good.
Yeah, by like 4 different people or 3 different people. All right, so this is the interim phase 3 data. And this is, of course, an Ad26 vectored spike, which is pre-fusion stabilized. And also they remove the furin cleavage site from the spike protein. They say to give a more stable product. And this was a randomized double-blind placebo-controlled trial, 18 years of age and older, of course, for safety and efficacy of this single-shot vaccine. Endpoints: moderate and severe disease at days 14 and 28. 43,000 participants, 8 countries, 34% of the people over 60, and 44% were in the U.S. Sorry for the numbers, Kathy.
No, it's okay. You're doing fine.
74% of the people in the U.S. are white Caucasian, 15% Hispanic and or Latinx, 13% Black African American, 6% Asian, 1% Native American. And
they comment that pretty much reflects the normal demographics of the population.
41% had comorbidities that are, you know, thought to be associated with whether you get severe COVID or not. And so what are the numbers? 85% efficacy against severe COVID-19 globally. And that includes South Africa, where most of the isolates are B1.351. That's pretty cool. Complete 100% protection against hospitalization and death. That's something that gets overlooked, you know, when you look at the numbers. It's going to prevent hospitalization and death. So my take is these vaccines are very good at preventing very severe COVID. And the mild stuff, the meh, as Rich would say, you know, it happens, but it doesn't really matter because it's meh.
Categorizing the different outcomes, I have found confusing. And so I guess it's probably confusing for others as well, because there's a difference between hospitalization and death on the one hand, or severe COVID on the other hand, or moderate to severe on the other hand, or asymptomatic on the other hand. So a vaccine that is good might completely prevent hospitalization, but not necessarily prevent you from getting some kind of sick, but maybe be better at preventing you from getting really sick, and not quite so good at making you get sick at all. And the numbers really reflect this. 100% keeps you, 100% of the people are out of the hospital not dying, 85% of efficacy against severe, 66% efficacy globally against moderate to severe. So if you count in the moderates as well, it's you get a lower number. So those distinctions, the wording and the distinctions are important, and they, in the slide presentation, give you a really nice, clear definition of what they mean by moderate, severe, etc.
Yeah, and the other thing to be really aware of sort of piggybacking off what Rich just said, is that this is something that you need to think about when you're thinking about some of these different vaccine trials. They have not all done the definitions of those groups in the same way. So in this study, the moderate group was you had to have 2 symptoms and then be PCR positive. In both Moderna and Pfizer, you had to have 1 symptom and be PCR positive. And so, the definition is going to be slightly different. There are other differences as well that I think we're going to talk about between some of the different trials. And so that's something that makes it very challenging to do a head-to-head comparison of any of these vaccine trials.
Not that you're going to have a choice ever, but if you'd like to know, right, that's important. But no one's going to say, hey, which one do you want? You get what they
this
have.
is what people are doing. They're saying, oh, well, this one's 95% effective. This one's only 80% effective. I want the 95. Well, you can't necessarily compare those numbers.
No, I don't think you can. I think as more of these results come out, we're learning you cannot compare the numbers. Because remember, AstraZeneca, also an adenovirus vector, albeit a different adenovirus, they decided not to distribute their vaccine in South Africa. They said it didn't work. And this one is working, apparently. So I don't know that that's just a vaccine difference. It may be something else. Who knows? So, I think you should take the vaccine that's in front of you.
Yep.
Yeah. It's like Daniel says, never miss an opportunity to vaccinate.
Yep. I know of individuals who really don't have any scientific background, but are, prowling through the internet trying to make a decision as to which vaccine they want, which to me is crazy given how hard it is to get any vaccine at all.
Right.
Mm-hmm.
If you have an opportunity, just take it. The time you spend waiting for what you think is a better vaccine to come down is time that you're exposed to getting disease, which is the worst thing that can happen.
Would you agree that you should not throw away your shot?
Yes, I would agree. That's an episode title.
I would not have gotten that a month ago. I finally watched it after all these years. And that was actually, I think that's the best song in the whole show. Yeah, first one.
Yeah.
And also supposedly on the Sunday morning news shows, Tony Fauci said, he would get this one. He would get whatever one is offered to him.
I think they're all good.
Same kind of thing. I
think they're all good. All these percentages that Rich gave you, they're similar according to age, comorbidities, sex, race, ethnicity. Protection begins at day 14. They have a nice graph showing the split in the number of cases between vaccine and placebo. It's starting at day 14. Cool thing, you can store this for 3 months in the refrigerator. That's really neat. At J&J, I heard today that Merck is going to help them manufacture this vaccine. They're going to make 100 million doses in the next 6 months. And this is cool. They're planning another study to see if a second dose would be better. Children 0 to 17 years of age, pregnant women, and immunocompromised patients. So that's all we need. We need all of that, and that's great that they're going to do that.
In terms of how these things work, because people may be interested in this, I want to give you my ideas. You can argue with me, okay? Because the vaccines that are available in the U.S. now are the messenger RNA vaccines in a lipid nanoparticle, okay? So it delivers a messenger RNA to the cells, and that's translated into the spike protein in your body, okay? This is a recombinant virus, adenovirus 26, expressing a spike protein, which is essentially the same thing. Because it gets into a cell, you make the messenger RNA, you make the spike protein. To me, the difference is, and this impacts on the storage and all that kind of stuff, but the difference is you don't synthesize this, you grow it, okay? And so in terms of manufacture, to me, that's a huge difference, to actually grow the product and purify it rather than to transcribe it at all and then package it in the lipid nanoparticle. And the other difference is how it gets in. In one case, you're taking up a lipid nanoparticle. In the other case, you're using the adenovirus particle to deliver the genetic material that's going to make the spike protein. But beyond that, in terms of what happens in the cell, I don't see any difference. They're ultimately just making spike protein in the cell.
Making spike. We're making spike.
I'm just about to put in an image that I saw. I think I saw it on Twitter. It's a Wall Street Journal diagram that does a pretty nice job of showing the difference between the two, the mRNA vaccines and the this newest one. But of course, I can't talk and do it at the same time. So, moments.
They also, of course, report on adverse events. And I was looking through these earlier, and in general, there are more in the placebo group than the vaccinated group.
All these things are extraordinarily safe.
It's amazing. Any medically attended adverse event, 400 in the placebo, 300 in the vaccine group. Serious adverse events, 137 in the placebo, 90 in the vaccine. Deaths, 16 in the placebo, 3 in the vaccine group. That's really quite remarkable.
And yet, I hear people tell me, I'm not going to take such and such a vaccine because somebody took it and died.
Yeah, no. These are fine.
Where do they get that stuff?
Or that, we don't have years and years of data about the mRNA vaccines, but there are lots of years of mRNA trials for cancer vaccines that, didn't go all the way to a full vaccine for potentially, I guess, other reasons, but we have lots of safety data on that.
Yeah, we have mRNA data, mRNA safety data since 2013 in clinical trials for other types of vaccines that just haven't made it all the way through for rabies and CMV. I feel like those slides I've been using quite frequently in talks lately.
Janssen/Johnson & Johnson points out that while this trial on this particular vaccine is 40,000 individuals, they have a bunch of other vaccines that have undergone trials so that they can say that this platform has been tested in 200,000 people.
They also have an addendum which contains data submitted after the original submission, which was a while ago, and this covers couple of different points. One is data on asymptomatic infection. In a preliminary analysis of asymptomatic or undetected infection based on an assessment of N protein seroconversion, right? Not spike. So N would be based on an infection. 2,600 individuals on day 71, 50 asymptomatic or undetected in the placebo, 18 in the vaccine group, which would be an efficacy of about 65%. So it's just looking to see if we can pick up an effect on infection, not the primary and secondary endpoints that we're looking at. So the implication is some ability to prevent infection. They looked in regions with variants, South Africa, 95% of the sample sequence, that's 86 out of 91, were B1.351, so it's predominating down there. Vaccine efficacy against severe and critical disease, 73% 14 days after vaccination, increasing to 82% 28 days. Also seen on mortality. All the deaths occurred in the placebo group in South Africa. So the vaccine, despite it being a different variant there, it still protects against death and hospitalization.
Now, that particular South African variant, is that the one that supposedly has a decreased response to immune serum or convalescent serum?
Correct.
Yeah, that's the one that has decrease in ability to be neutralized.
By sera, yeah.
And yet there's no difference in the efficacy of this vaccine in that environment relative to other countries.
That's right.
So that's really great news.
I mean, that's the number you want. You want to know how it prevents death and hospitalization. I mean, the neutralization doesn't tell you that. It just tells you neutralization, right?
Exactly.
Right. And so that's really great news.
And so I don't remember, what was the basis for the pulling of the AstraZeneca? Was it this kind of data, or was it just the neutralization data?
No, it wasn't neutralization.
No, I think they were doing a trial, right? And they were seeing an equal number of COVID cases in the placebo and the vaccine at a time when I think they just had 20 cases in each arm. Mm-hmm. They said, "Oops, that's not good enough."
Yeah, it was. I think it was early, so it's too bad.
Now, the one difference, I mean, yes, it's an adenovirus vaccine in though a different vector, but beyond that, the one difference, if I understand it correctly, between the AstraZeneca vaccine and the mRNA vaccines or the Ad26 vaccine is that the AstraZeneca vaccine is not a pre-fusion stabilized spike, correct? I believe that's what Jason told us.
Yeah, that's right. He did say that.
Right.
Okay.
Right.
Now, that's just, I don't know whether that makes a difference, but that's what comes to mind for me.
In Brazil, where they have 69% of sequence samples, 86 out of 124 are the P.2 lineage. They say the efficacy is still high. It's similar to that observed in the U.S., where D614G is the main variant, and so they say it's even working in Brazil. This is from these supplementary data, and they also have supplementary data on hospitalization, but basically supporting this idea that it's really quite high. Preventing hospitalization is huge, over 90% in death. So I think this is all great news and a lot of doses available. I think this week they're starting to distribute them. It's great. By the way, this platform is the same that's been used for an Ebola virus vaccine, which has been approved by the European Commission. It's an interesting prime boost approach where you get Ad26-ZBOV spike first, and then you get a modified Vaccinia Ankara as the boost. So that's approved. So this vector has been already approved for another use. Very cool. Then there's, as I said, another slide deck where they have collated the vSafe data. People who get vaccines, you can get this app on your phone and report in what's going on. So that's Pfizer and Moderna mainly.
Right. And what's interesting is that if it gives you the number that have been given as of, I think it was mid-February, total of 55 million, and less than 4 million of people have gone and done this app. So it's only about 7%. But then from that, they have a bunch of things that they can look at. And the main thing is that these are self-reported and they can help generate hypotheses about potential vaccine safety concerns. But there's a lot of limitations. There's the reporting bias of only the 7% that decided to go forward with this.
Yep.
There's not a control group that's unvaccinated, and this is not designed to assess causality. But it does have the advantage that it can draw from a huge population nationally pretty quickly and could help detect very rare events. And it's publicly available. So it's nice.
It strikes me as very similar in concept to VAERS, only easier to VAERS being the vaccine. What is it?
That's what it is. It is.
vSafe is VAERS?
I thought so. It was the very next slide, but maybe
I have the impression that vSafe is
a feed into VAERS.
Yeah, yes. I think that it feeds into VAERS.
Yeah. Okay.
It's not clear to me if everything that's reported in vSafe is as adverse as what qualifies for VAERS.
Okay.
Yeah, I think this vSafe was made so that when you're there sitting there for 15 minutes, there's big signs, download this app and register right now, and then they get you. That's what I did. And I answered every time they asked, and I thought it was cool to be able to do that. So I don't know. It's too bad only 7%. But I do think it's a separate batch of data. It's collected differently, right?
Well, if it all goes into VAERS, it's probably flagged as having come from, at least that's the way I do it.
So VAERS is a little different. I mean, VAERS I don't think has an app. You have to go online I could be wrong, but it says, report an event on using the online form or a new downloadable PDF. So it seems like a different. And then they have separate data for VAERS compared to the vSafe, right, which are far smaller.
Yeah, vSafe call center conducts active telephone follow-up on a clinically important event and takes a VAERS report if appropriate.
Yes, which is a different thing, right?
So that's on their slide 5 in that link.
Yeah. Anyway, I think it's what I thought is interesting is that pregnancies reported. So 30,000 pregnancies in people who have been vaccinated. That could be useful, right? Mm-hmm. Although, as you say, it's not technically great data because it's self-reporting, but it can give you trends that you can test. So I think that's cool.
Right. And so on that same slide, they say that then the pregnancy registry team conducts outreach to assess eligibility for registry and obtain consent for enrollment and follow-up.
Yep.
So it's all good that way.
Yeah, I enjoyed doing it. It was pretty easy. Get a little text. You click the link. They ask you a bunch of questions. Boom, you're done. And then after so many days, they stop bothering you. I feel like I contributed something. So go do it. Mm-hmm. So anyway, there are a lot of good documents associated with this. Also with the other ones, there were many documents as well. And if you're so inclined, go check them out. And interestingly, people pour over these data. There have been a number of preprints submitted where they go back to the Pfizer, Moderna data and say, hey, look, we found this or we found that. So it'll be interesting to see what comes out of this as well. All right, let's do some picks. Brianne, what do you have?
So I have a short YouTube video. It's about 8 minutes long. That is by a group called Science IRL, Science in Real Life. And this video is called How T Cells See SARS-CoV-2. It's actually How T Cells See COVID-19, but I couldn't say that. So I had to,
Good for you.
I had to change the name.
Good for you.
So basically, there is a person who makes these YouTube videos who interviews a scientist who works on the T cell response to SARS-CoV-2, and they explain all of the different aspects of SARS-CoV-2 with little images, not Janet Iwasa level images, but still pretty great images. And I think it's a nice explainer, especially for people who are less up on T cell biology.
It's very nice. I had a look at it. It's good.
Mm-hmm. I did too.
Rich, what do you have?
So I have, I use a Windows computer running, Windows. And so it's got this thing that pops up when you log in that's basically Windows clickbait that's trying to get you with cute images to use Bing, their browser. That's the way I see it. But some of the images are awesome. And this one really caught my attention. And I tried to recover this in a fashion that wouldn't use Bing, but I couldn't get the picture with the nice explanation. It's called Ice Bubbles in Lago Bianco, Switzerland. And the picture is just, if looking at this, it looks like modern art. You would never know what it is. It looks like a bunch of stacked, very three-dimensional white discs with some shadows and a dark blue background. And what it is methane gas produced by bacteria through decay at the bottom of this lake that gets trapped in bubbles as the lake freezes. All right. And if you click through this, you can get a number of other related images. And it's just a beautiful picture. And I thought not only a beautiful picture, but the science of how it is that it arises, I thought was interesting as well.
Very pretty. Amazing shots. Yep. Kathy, what do you have?
I have something that was sent by a friend of mine, Mary, and it's an article about, I think it's a state senator, a Republican senator in the state of Washington, where he's proposed a couple of things. And one is to go ahead and have spring break be right when spring break would be, but have it be 2 months long, and then take up with the last 2 months of school when potentially the SARS-CoV-2 cases would be lower, and people could be outside. And so that's part of it. But the other part that really catches your eye is these tents, person-sized tents of half of the band kids in the auditorium, each one standing in their own tent. And they're this lime green color. And so I already thought about, well, if we did this and we were singing, maybe they would be white and that would be less glaring. But anyway, it just shows that them playing their band instruments inside these tents. And so it could be a way to go forward. In this instance, they have done that. And evidently morale is up and so forth. I showed it to my cousins on a Zoom, and they pointed out that one of them, was picturing how a sousaphone would be. and two of them play the trombone, and that also would not work very well unless you somehow sat down and turned the tent on its side. But anyway, it was just really pictures worth 1,000 words here.
These tents must have been specifically fabricated for this use, you think?
I think so. Yeah, it seems like it.
I can't think of any other way I'd want to use this.
Yeah, I can't either.
It's big enough to hold a music stand and a person and a saxophone or not a trombone.
A person who's standing up. You couldn't lay down and take a nap afterwards.
No, right.
Looks like they have taped windows on them, so they do look somewhat handmade. Yeah. I bet they get warm in there, though, right?
I don't know. Maybe.
There's probably no ventilation,
if
right?
If it's ripstop nylon, maybe it's breathable. If it's not coated, yeah, I don't know.
Interesting. All right, what do we call it? Ingenuity. Right. Mm-hmm. My story, since you guys went crazy on the Perseverance last week, my version is a story in MacRumors, which the original is in New Scientist, but you need a subscription. They have more writing there, but I don't have a subscription. Anyway, the Perseverance uses a computer processor found in 1998 iMacs made by Apple. It's called the PowerPC 750, and it's a single-core 233 megahertz processor with 6 million transistors. And today, there's more in your iPhone today in terms of processors. But they say this one was used, and I don't know how long ago they decided to design this thing. That may be part of it, right? Because it's such a long lead time. And they can't change it in between. But this one was built specifically to withstand temperatures between minus 67 and 257 degrees Fahrenheit. That's minus 55 to 125 Celsius. And it costs $200,000 because they probably only made two.
Yeah, it's a little more expensive than those old Macs were.
Yeah. That's really funny.
Tough. It's reliable and it's tough.
It's tough, and it's enough.
The justification.
I mean, that's all the processing they need for this, right? You don't need 16 billion. And the space shuttle used to run on an old IBM XT, I think, right, for many years. So you don't need that much. It sounds scary, but anyway, that's my contribution to the Perseverance arc.
Oh, and I want to just continue with that a little bit. Today's Science Times in the New York Times has a feature. It shows the parachute binary code and a couple of the other things that we talked about. And then the article also shows that there's one of the things they sent up was some pieces of a Mars meteorite that had landed on Earth. And that's there for them to do some calibrations on. And that same Mars meteorite has been to the ISS and back. So now this is your lead-in, Rich, to your ISS story.
Thank you very much. Thank you very much, because I needed that. For those of you who have followed our Kate Rubins arc, all right, Kate took a 7-hour spacewalk last Sunday. I'm sorry I didn't give a heads up on that, but she's going out again on Friday. So that'll be after this episode drops. They're putting together some supports for what will be an upgrade of the solar panels. So I highly recommend this. I just happened to be awake at 4:30 in the morning, knew that this was on, turned it on, and I got sucked in, and I watched the whole spacewalk.
7 hours you watched?
More than that, because I watched them depressurize and repressurize.
Wow, that's commitment, man.
Yeah, it's like it's better than a Warhol movie, though. It was great.
And that's I was just going to say that's the type 2 fun, as
type
Kate describes it. Yeah.
That's right, type 2 fun. It sounds like fun before. It seems like fun afterwards, but while you're doing it, it's a headache.
They're just building something with wrenches and stuff, right, out there.
Well, so they're building these big triangular struts that fit on the side of a big cylinder that holds the arrays. And of course, as you might imagine, it's all pre-designed, it all figured out. And so they had to carry out these struts unassembled as linear individual struts in a couple of big, what they called strut bags, and then fasten them down to the station and pull out one bit at a time and bolt them together. They've all got bolts that are already affixed through the holes. I've seen this kind of thing before. They're so that they can't come out, but they can screw into something else, because you don't want bolts floating around, right? You don't want anything floating around. And so they bolt those together and they assemble this triangular thing and then affix that to the station. What I was unclear on, I actually wrote Kate an email with a bunch of questions. I don't expect necessarily an answer. She's got other things to do than answer my silly questions. But they were screwing these things into, bolting these things into holes that were already on the space station. One of them was a mount that was on the side of the cylinder that was clearly constructed for this thing, and I wonder if that wasn't put up there previously. But the other looked at was as if it was on some sort of pre-existing thing, and I'm wondering, like, how did that hole get there? Or is that another mount that they added and blah, blah, blah. At any rate, they bolted it to the side of this cylinder, and later this year, the panels, they're going to finish the job on Friday. They didn't quite finish, but they finished everything they were scheduled to do. And later this year, the panels are going to arrive, and they're going to set up the panels. This is Kate's third spacewalk. She has spent 19 hours in space. Okay, she's headed for her fourth.
All right, I have a question which may not apply to them because they're young. I couldn't last 7 hours without going to the bathroom. So they have something in their suit that they can...
I think they wear diapers.
They wear diapers? Okay.
I think so. I wouldn't be drinking a lot of coffee before I went out for a spacewalk.
Not like Alan Shepard, huh?
No.
Apparently he drank a lot of coffee that morning, and his launch was delayed, and they said, just pee in your suit.
Yep.
All right, that is TWiV 726. Show notes at microbe.tv/TWiV. Questions and comments, TWiV@microbe.tv. If you want to support us, we'd love it. Join the throngs, well, not throngs, but join many people who support us, for which we're grateful. Microbe.tv/contribute. Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Brianne Barker is at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
Rich Condit is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough. And I'm just thinking that if these podcasts are any longer, I'd probably have to wear a diaper.
Sorry about that.
TMI.
TMI. Yeah, TMI.
All right. Always a good time.
I'm Vincent Racaniello. You can find me at virology.blog. You ain't going on a spacewalk, Rich. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back in a few days. Another TWiV is viral.